Welcome back for part 3 of 4. So in this one, we're going to look at what happens when meiosis doesn't work correctly. You've kind of introduced yourself to this topic with LabNet last week. So we're going to be talking about, having too many chromosomes or having too few chromosomes overall. Okay. Now, in general, any kind of large-scale changes in chromosomal number is going to lead to a spontaneous abortion. Basically, it is not a viable a viable fetus at that point in time, and instead of moving forward, there are internal systems to be able to go, okay, shut it down. It's not going to work. All right. But there are a number of them that are viable, and we see a lot of disorders being caused, particularly in mental and physical development, by changes in numbers of overall chromosomes. Okay. Now, in general, plants generate this much better. They often have, instead of being haploid or diploid, they could be triploid or even quadruploid. They have complete copies of their genetic information. And as to why plants tolerate this better than animals do, we're not really sure. There must be some additional mechanisms that plants have evolved into that animals don't have. Now, anything that changes overall chromosome number is going to be this idea of non-disjunction. So what we normally see is, separation of homologous chromosomes during meiosis. Non-disjunction is going to change that by not having normal separation occur. So in general, what this means is that one of the gametes is going to get 2 copies, and another one of these gametes will receive no copy of that particular gene. Now, if we follow through with the events of what's going on here, like we've got non-disjunction here in meiosis 1, and we've got normal on this side. So in this case, we've got normal separation on first chromosome. Our second chromosome, the homologous pair does not get separated. So when we get into meiosis 2, we've got one cell that has 2 copies of that particular chromosome and another cell that doesn't have any at all. But non-disjunction can also happen in meiosis 2. So we're going to see 2 cells here potentially that are going to have normal numbers, and we're going to have 1 cell here that's going to have that duplication of chromosome. So depending on when it happens, we get a variety of different types of gametes out. If it happens during meiosis 1, we're going to see 2 types of gametes form. One that are going to have an extra copy of the chromosome, and ones that are going to have 1 less copy of that particular chromosome. Now, if it happens during meiosis 2, we're only going to get 1 out of the 4 cells that have either an extra chromosome or 1 less. And we'll get half the gametes still having a normal number of chromosomes in the end. Now, when it happens, in the end, doesn't necessarily change the overall outcome of that particular union of a gamete that has the wrong number of chromosomes with one that has a normal number. Now, aneuploidy is the result of fertilization of gametes that have non-disjunction. So they're going to be this broad class of fertilizations that have either an extra number or a fewer than normal number of chromosomes. So offspring with this condition will always have an abnormal number, either above or below. They might be monozymatic, which means they've only got 1 copy of a particular chromosome. And in general, this is going to be a embryo-lethal condition. There are a few rare instances, one of which we'll touch on by the end of this lecture, where the zygote is viable, at least to mature. Trisomy is the other option, where we have 3 copies of a particular chromosome. And generally, this is also a instance where it's going to be embryo lethal. But there are a few more cases where this produces a viable embryo that can reach adulthood. Now, polyploidy is a condition where we multiply the number of chromosomes overall. somehow all of the chromosomes don't properly split, and we can see triploidy or tetraploidy in the end. Quadruploidy was an old term for it that I used earlier. And this is most common in plants, but we have yet to come across an animal that has this condition. Plants, somehow this happened early on in their evolution and stuck around in certain plant species under those conditions. Not sure how, but it works for them, so we've got a mystery to solve in this case. And polyploidies are pretty much normal in general in most cases than autaploidies. So having a complete extra set of your chromosomes seems to be less of an impediment than having fewer than the right number of chromosomes in the end. Now, there's also instances where the chromosomes themselves are going to change overall shape. And we can break these down into 4 basic types of changes. And we're going to go through each one of these in the end. We have deletion. So this is going to be actually cutting out a portion of the chromosome and leading to a shorter chromosome overall. We're going to see duplication. Something unusual happens and these genes kind of get extended again. So we end up with a larger chromosome that have replicate information inside of it. Inversion. So you might remember that we read our DNA from the 5' to 3' direction. Well, what happens here is that normal 5' to 3' reading frame and its anti-parallel strand that runs with it, well, they reverse. What was 5' to 3' flips, now becomes the anti-parallel strand that's running 3' to 5'. And what used to be our anti-parallel strand is now our 5' to 3'. So this can completely change the sequence order of the nucleic acids inside of DNA. And then lastly, we have translocation. And this is going to be where a portion of the chromosome somehow gets moved into another chromosome. And in some cases, this isn't as bad as it sounds. It's possible for this to be completely what's quote unquote silent, not affecting the individual, but it all depends on where these breaks happen. If you get a full gene or if you get a partial gene, if that dramatically changes everything. So let's look at deletion. Okay, deletion being a simple removal of a portion of the chromosome. So this usually happens during S phase. Something happens during the duplication of DNA and it doesn't quite work out. We lose a portion of it in the end. We end up with a shorter overall chromosome and we're missing some number of genes within that segment. Duplication is another error during S phase. Somehow this segment gets reset and we start duplicating we start copying it again. So we end up with a chromosome that's longer, that has multiple copies of particular alleles within it. Again, this might not be that detrimental in the end. As you can imagine, deletion, not having a gene, probably not as bad as having some duplication. Inversion. This is where the reading frame changes entirely. So normally it's B, C, D. We break both strands of the DNA. We flip it. So this portion here ends up down here, and this portion here ends up down here. So basically our 5' and 5' to 3' strand and our 3' to 5' strand reverse in this case. And this has all kinds of interesting effects, usually being pretty bad for the organism involved. Though if it happens over a short period, this is a type of mutation that can actually lead to some advantages. And then translocation is not quite the same as crossing over, but you can imagine it happens kind of similarly. Maybe, instead of getting all of the normal genes involved or happening across two non-homologous chromosomes, crossing over that is, we can get the swapping of genes from one to another. And if this happens at a point where we're not coding any kind of active gene, this may not actually be a detriment to the cell in which it happens. So let's take a look at least a little closer at a few disorders that happen because of these chromosomal alterations. So a lot of these are pretty serious in the end. Some of our audiploidies are going to upset the overall genetic balance more, leading to greater deficits in physical and mental development in the end. And ultimately, very few individuals are going to survive to birth and beyond when we have an abnormal number of chromosomes. So those that do survive often have a defined set of symptoms, which can ultimately be called a syndrome. And they are always characteristic of that particular type of defect. So perhaps the one that is most commonly known is Down syndrome, or a trisomy of chromosome 21. So we have our karyotype here. We can see chromosomes 1 through 20 have a single pair of each. 21 has 3 copies. 22 has 2, and then we've got the normal XX combination that produces a female child in this case. This affects about 1 in 830 children overall, and it seems to be linked to older mothers giving birth to children. Not exactly sure why. There have been theories that there is something that happens during meiosis in older parents that lead to non-disjunction happening more readily. I've even read a theory that it's not just female age that is involved in this, but also male age. But it seems to be most strongly linked with female age in this case. There are other mechanisms that might be in play here, just like, well, both men and women have, kind of internal checks for if their gametes have an abnormal number of chromosomes. And maybe somehow it just sneaks through on the female side more than on the male side. We don't really understand what's going on in this case. Now let's talk about audiploidy in the sex chromosomes and kind of multiple X and Ys or not having them in the end, because kind of an interesting subtopic in the end. So this is actually a group that can actually be relatively survivable in most cases. Okay, so Klinefelter syndrome is a case when men have 2 X chromosomes in their cells. Okay, we see this is about 1 in 1,000 males, and we don't really see any kind of overall syndrome in the end. There's some linkage to being taller, being a little thinner, but other than that, perfectly normal when you get down to it. Now, if we have a audiploidy that leads to 3 X's in females, again, same frequency, well, they have no physical characteristics, kind of similar to the men, but there does seem to be some kind of a linkage between mental development and having a triple X chromosome. This might be impartially kind of associated with, remember Barr bodies? When we talked about them, we had that inactivation of one of the X chromosomes. Well, this doesn't always happen from what I know in this condition. So there might be some additional interactions between different versions of a particular gene that can change the overall very complex development of the brain and might lead to some of the developmental problems that we see. Now, the one monosomy that we have that is very survivable is Turner syndrome. So this is for women who have only 1 X chromosome. They're unable to have children, but other than that, they seem pretty normal. They're a little smaller. they're not going to develop secondary sexual characteristics, and if they do, they tend to be very minimal. No breast development, not a lot of pubic hair development in the end, and it's one of the few cases where monosomy is at least documented survivable in humans. We don't know of any other conditions that really allow that. Now, let's get into a few that are leading to like the changes in the chromosome that we talked about before. Deletions, insertions, duplication, things along those lines. So this cri du chat, this cry of the cat, is going to be caused by a deletion inside of chromosome 5. And this leads to a kind of high-pitched meowing cry in these infants. And, is unfortunately not a very kind syndrome, okay? It's survivable for a short period of time, but they generally don't make it past early childhood because of the number of both intellectual disabilities they have, but also physical disabilities because of the missing chromosomes on 5. Now, this can also be linked to certain cancers, at least in theory. So, myelogenous leukemia that actually affects the bone marrow in particular, can be caused by a translocation of chromosomes. So I've got an example of that here. So if chromosome 9 and chromosome 22 somehow go through this process of translocation where a portion of chromosome 9 is swapped for a portion of chromosome 22, there's a break here of some kind of key protein needed for normal blood cell development. In this case, the bone marrow is not going to be able to produce the normal cells that it can, and they turn cancerous. We see this blood-borne cancer, this leukemia, form under these conditions.